I'm Christy Taylor, and I lead the computational molecular design team here in Chesterfield in plant biotech in the data science and analytics group. And I'm very excited to introduce Mohammed Al-Quraishi, who is an assistant professor at Columbia in the Department of Systems Biology. The Al-Quraishi lab focuses on two biological perspectives, the molecular and the system levels. And on the molecular side, his lab develops machine learning models for predicting protein structure, function, machine learning models for predicting protein ligand interactions, and learn representations of proteins and proteomics. Dr. Al-Quraishi holds an undergraduate degree in biology, computer science, and mathematics, and he earned a master's degree in statistics and a PhD in genetics from Stanford. He joined the faculty at Columbia in 2020, and on a personal note, here at Bayer, we're part of the AlphaFold consortium, and Mohammed we collaborate a lot with his lab. So I'm very excited that he came to talk to us today. And I'll turn it over to him. Thank you.
I guess I don't have a clicker, right? Or do I? Okay. Okay, no worries. Thank you very much for the invitation. It's a pleasure to be here. Yeah, thanks to Bayer for the support of the AlphaFold Consortium, which has really been transformative to us, as I'll talk a little bit about during the talk. So I guess next slide. Okay. So what I thought I'll do today is actually give you a bit of a kind of an overview of status update of the field of protein structure prediction and sort of related problems, right? Things having to do with, as I'll talk about, conformational ensembles, predicting the impact of mutations, problems that are still at the frontier of what we currently do in sort of machine learning of molecular systems, particularly as they pertain to proteins. So I actually won't talk a ton about our own research. This will really be more of a kind of a status update, because I thought that might be more interesting to this crowd. But I'll point out certain pieces that do have to do with our work as I go along. So next slide, please. So I'll basically sort of, like I said, kind of walk you through a number of topics. But before I do that, let me just kind of give a very, very high-level overview, quick overview of AlphaFold 2, which was really a sort of seminal development in the field, the system that I would argue kind of, at least when it comes to kind of globular single-domain proteins that have well-defined kind of evolutionary sequences because they're natural proteins. At least for that problem, I think I would argue this system has to solve the protein structure prediction problem. So at a high level, what AlphaFold 2 does, this is on kind of on the left, is it takes a critical piece of information. That's a multiple sequence alignment. That's essentially a kind of an evolutionary history of the protein of interest, potentially dating back millions of years. And that multiple sequence alignment is then fed through a kind of a series of neural networks. And I won't go into the details of it right now. That transform that representation of the evolutionary history into a kind of a structural object, an object that describes where atoms are with respect to one another, and also where the site, particularly the side chain orientations of the amino acid chain. But key to this procedure, and then I'll come back to this later on, is this multiple sequence alignment. Because essentially what this network does is it converts information about how residues co-evolve across evolutionary time into structural information, into information about the proximity of those residues in 3D space. And it's that conversion from evolution to structure that's kind of the key enabling, I would say, sort of inductive bias in AlphaFold 2, and when that when that prior kind of breaks down, as I'll talk about in some instances, AlphaFold doesn't work very well, and I think that will drive us to have to develop new technology to be able to tackle these problems. Next slide. All right, so I'll sort of, like I said, walk through kind of 5 main areas that I think are currently at the frontier of where we are. And I think maybe the first 3 will be, or the first 2 will be areas where there's, I would say, sort of more progress has been made, and then the last 2 are sort of still kind of the frontier of where we are. So let's start with monomers. Next slide. This is more to kind of situate you of kind of the critical experiment that really sort of defined AlphaFold 2. So this was Cas14. It's been a few years now, and the focus here was on yeah. You could go ahead and sort of progress, maybe progress a couple more slides, one more. let's stop here. Okay, thank you. So the focus here was on single apoproteins, so proteins that are unbound to anything, and this was kind of the debut of AlphaFold 2. And as you can see on the slide, the predictions that are in green versus the ground truth, which are in blue, are generally very high quality. The accuracy, I think, the median accuracy was like 2 angstroms, and something like 90% of the predicted structures had a prediction accuracy of less than 4 angstroms, right? So this is why I said, in some measure, this model really seemed to solve that well-defined kind of specific version of the protein structure prediction task, right? And again, this has been a few years now, so this is somewhat old news, but this really kind of set the stage for what was to come afterwards. Next slide. Now, already at this experiment, we begin to see maybe some cracks, some of the issues, some of the limitations that will sort of begin to kind of be a problem as we move to kind of use AlphaFold 2 in other contexts. So there was a prediction task at Cas14, this particular target, T1056, which happened to be a zinc ion binding protein. And as you can see in this figure, the prediction is actually quite good in the sense that there is high overlap, right, between the side chain atoms of the predicted and the experimental crystallographic structure. So on one level, this looks to be a success. Right? This is something where the model is actually behaving as you'd hope it does. But on the other side, and this is the limitation here, the fact that there is a zinc ion present in that pocket is not something that the AlphaFold is sort of has in its preview. It does not understand, it does not even know anything other than amino acids. So the fact that it's able to sort of place all these atoms in the right place is really more sort of a reflection of the fact that it's sort of seen these types of patterns in PDB, and it's able to almost sort of impute or kind of hallucinate what might be present there, even though it doesn't have a conception of a zinc ion. And this becomes a problem, obviously, when we think about other types of ions or other, say, ligands or small molecules, where we want the model to be responsive to their presence or absence. And in this instance, right, it isn't really. It's just sort of accidentally giving us the right answer, but not necessarily for the right reasons. And that's sort of the problem. Next slide. Another thing we sort of set out to do, and this is a group from the AlphaFold work, is to assess how well the model does when we essentially task it with generalizing to unseen regions of structure space. And I'll sort of walk you through this experiment kind of step by step. So the first thing we try to assess is, well, how well would the model work if it were trained on a subset of the PDB, right? So AlphaFold 2, when DeepMind developed it, was essentially trained on everything in the protein data bank. But one can sort of imagine this experiment, this thought experiment of training on a version from 10 or 20 or 30 years ago and asking, well, how well would it have done if we had done so? So what you're looking at here on this figure is, so first the color coding essentially corresponds to how much of the PDB is retained when we do this sort of experiment. So pink, kind of the top, is the entirety of the PDB, something on the order of 150,000 single domain structures, and kind of the light blue teal is essentially a fraction of a percentage point, something on the order of about 1,000 PDB chains. And then the curves that you see are essentially the performance of the model as a function of training time. That's what the x-axis is, measured using GDT, which is the y-axis. That's a particular metric that we use where 0 is, sorry, this is LDDT, where 0 is very poor and 1 is a perfect prediction. So the pink curve is essentially the best model, right? The model that's fully trained. And as you can see, that that sort of it cracks 0.8 at this figure that I'm showing here, which is reasonably high. And then if you go all the way down to the blue model, right, that was the one that's trained on like 1,000 structures, clearly there's a pretty substantial drop in accuracy. But what's remarkable, right, is if you look, for example, at the green curve, that's, I think, again, the 1.5%, that's on the order of 2,000 PDB structures, the model is actually pretty performant. The model is, in fact, achieves an LDDT here of 0.7, which is better than what AlphaFold 1 did just 2 years prior. Which was a state of the art at the time, right? So one way of looking at this, right, is to say that the architectural innovations introduced in AlphaFold 2 enabled it to essentially be 100 times more data efficient. Right? It was able to outperform AlphaFold 1, which was trained on 100,000 structures, by simply coming up with better neural network architectures that are able to exploit and leverage the data that's present in those 1,000 structures, right? And that really kind of highlights the importance of thinking carefully about how one constructs these models, right? And I think it also bodes well in a way to other tasks where we have a lot less data, for example, RNA structures or protein ligand interactions, where we can't, this is no longer a thought experiment, but it's sort of our reality, that really the only thing we have to work with are, much more limited data sets. Now, in this experiment that I'm showing here, the exclusion was done completely randomly. So you might argue that, yeah, you have less data, but the diversity is still unchanged, and then maybe the model is doing well because of that fact. So next slide. What we next do is essentially kind of stratify structure space using existing stratifications like CAF in this case, which sort of cluster structure, right, in a hierarchical manner from kind of class level divisions. So, alpha helical proteins, beta sheet dominated proteins, and so on, all the way down to sort of finer grained distinctions called topologies in their terminology. Next slide. So if we first look at the topology level, that's the lowest level, right? So this splits the PDB in maybe about 100, I think, odd topologies. So you can think of 100 different structural clusters. And when we do this, again, you see a drop in accuracy. So now the percentage that the color corresponds to is not the percent of number of structures retained. This is the number of topologies retained, right? And then we test on the remaining. So even though there is a drop in accuracy when you only look at something like 5% of the topologies, but already with 10%, again we're outperforming the kind of the first AlphaFold, indicating that the model is able to generalize at least at the level of topologies. Now, as we progress in the hierarchy, so next slide, to architectures, and then furthermore, next slide, to classes, we actually see this more or less remain the same. And this was really very, very striking and shocking to me, at least. The fact that if you essentially train on alpha helical proteins and then assess the model performance on beta sheet proteins or vice versa, you again have a model that's comparable to something AlphaFold 1. And just to illustrate this visually, next slide, I'll show some actual examples here. So on the top row is a model that's essentially trained on just largely alpha helical proteins. And in orange is the ground truth structure. And so we're assessing that model now. In magenta is the prediction from that model. And you see it's not surprisingly doing quite well because it was trained on alpha helical proteins. But then in green now is the model trained on the beta sheet dominated proteins tested on this protein. And you see here that while it hallucinates a couple of strands, the structure is not entirely terrible, right? And the converse is on the bottom, where now you have a protein that's largely comprised of beta sheets, and you see that the green prediction is quite good, not surprising, because that was trained on beta sheets. But the magenta prediction, right, that's a model that's essentially only sees alpha helices, and is able to more or less recover the geometry of the protein that's dominated by beta sheets. And what's interesting there is the secondary structure is not quite good enough for something like PyMol or DSSP to actually recover the hydrogen bonding networks, so you can actually see the strands. That's not there. But the alpha trace, so roughly the kind of overall geometry, is well recovered, and that's what's really kind of, I think, surprising here. So the model is sort of able to make rather sort of great feats of generalization that I think is not maybe one that one would expect just from kind of standard machine learning intuition, where these things tend to be very fragile and they start to break down as soon as you move away from sort of your training set distribution. Next slide. All right, so that's it for monomers. I'll try to go more quickly through the subsequent sections, because I know I went to the stage, but hopefully now we can kind of move fast. So all right, so let's get to mutation, which is the next piece, guys. So this is obviously of interest for a lot of people, because what we want to be able to do is we want to be able to predict, say, for example, how pathogenic variants, right, in the human population impact structure, and not simply structure that's sort of fully disrupted where the protein is no longer folded, but structure that is changed in some specific way that perhaps makes the protein react differently, either in the natural context or in response to, say, a drug when we're trying to modulate it. And the fundamental issue here, I think, is something I alluded to in the very beginning, right? That these methods are based on multiple sequence alignments, and that allows them to look at the evolutionary history, but that also essentially makes them average out all that variation to predict a structure. So providing a sequence that's mutant in a structurally disruptive way is almost adversarial to the way this model was trained, because the model was trained to essentially ignore those types of perturbations, right? And I think that there was and is some hope that protein language models, like ESM, if some of you may have heard of these, could actually sort of address this problem. Next slide. And just to kind of situate everyone, so ESMFold, which was a model that essentially took a protein language model called ESM2. This is a model that was trained on hundreds of millions of protein sequences in an unsupervised way, just to simply recover the amino acid identities, a bit like ChatGPT was trained on natural language. This was fused to OpenFold to kind of now yield this sort of entire system end to end that essentially acts like AlphaFold 2, but replaces the MSA component with this language model component. Next slide. This is it's a great model. It's very fast, and that actually allowed Meta to essentially make predictions across all the known protein universe, and yielded a sort of valuable data set actually for potentially kind of understanding the topology of structures, particularly in less well characterized organisms. Next slide. But when it came down to the question of whether it sort of addresses this challenge of being able to predict the structural impact of mutations, unfortunately didn't do very well. So this is a bit of a busy slide, but I'll try to walk you through it. So here we have Cas15. So this was a sequence experiment happened 2 years ago now, where again the impact, the focus here was on predicting structures. The curves in blue are ones that are coming from multiple sequence alignments. So those are kind of standard models, and then the ones in red are models that use these language models, right? So the ones that are kind of working without MSAs. So not surprising on the left. So this is what the x-axis essentially spans is the depth of the MSA. So how well characterized in sequence space is that protein? And on the left, all models do well. So high is good here, and that's perhaps not surprising. But on the right, as you go further and further in the less shallow MSA regime, you're dealing with proteins that don't have well characterized evolutionary histories. And what you see is that both types of models do worse, but surprisingly, the language model-based ones, the ones in red, actually seem to get even worse more quickly than the MSA-based ones, suggesting that these models aren't really able to kind of deal with the fact that you are that you're sort of operating in this regime where we have very shallow MSAs essentially. So this is a bit of a negative result. We're currently working on a model that we hope will sort of overcome this, and that's something one of the kind of the main thrusts that we're focusing on in OpenFold, and we have pretty promising results so far. But at least in language model-based approaches, don't really seem to move the needle very much. That's next slide. I'm thinking that maybe perhaps for the sake of, well, let's see. Let me try to go on through multiple, but I might skip confirmations and get to protein ligand right after. Let's skip this slide if you don't mind. So next slide, please. This might be a bit tricky. Next slide, please. Next slide, please. Okay, let me pause here for a moment. So at a high level, right, people were able to use AlphaFold to predict complexes of proteins. So that was kind of an early quick win. And a sort of a defining aspect of this fact is that if you look at the kind of the 5 bottom models here on this figure, these were all models. So just to set you up a bit, the green bar was essentially the pre-AlphaFold 2 baseline. And again here, 0 is bad, 1 is good. So we want those bars to be as long as possible for assessing the quality of predicting complexes. Green here is the baseline, and then the 4 other curves on the bottom essentially of this figure are ones in which the authors just use AlphaFold multimer. Just use AlphaFold 2 without doing any retraining. So just the original model, but just trying to predict complexes with it, right? And you see already we see substantial gains over the green bar, which was the kind of the baseline. But then in Cyan, that's the very top, DeepMind went and essentially retained AlphaFold 2 on multivariate complexes and tried to tackle the complex problem, right? The complex prediction problem. And what was really surprising here is that just by that simple retraining, you see substantial gains in accuracy. And part of the reason why I highlight this is because in the case of the AlphaFold 2 code base that was initially released, there was no training code. So it wasn't possible for anybody in the public to actually train this model on new data, including multivariate data, to be able to do what DeepMind did. So part of the motivation for us with OpenBind, with OpenFold and more broadly, rather, it was to essentially allow people to retrain a variant of AlphaFold 2, not just on multivariate data, but also on private data of all sorts, including potentially pharmaceutical data on protein complexes or protein small molecule interactions, which might be of relevance for specific drug discovery programs. And I think this kind of figure really illustrates just the value of, even without introducing any architectural innovations, just retraining the model on private data or on different types of data, what sort of gains you may be able to achieve. Okay, next slide, please. So next slide. Let's get to the, I'm going to skip this section, so let's get to the kind of the outline again, if you don't mind. So keep moving forward. Yeah. Let's go ahead and actually move to the small molecule ligands. So skip through until we get to step 5. Just so I want to get to that because I think that's valuable. Yeah, let's stop here. So I'll end with this section. Sorry, yeah, stop right there. So I think of relevance to this guidance, probably when I make sure we get to this section, is protein small molecule interactions, guys. And I started in the very beginning mentioning the kind of zinc ion story and how that leaves out a kind of a key aspect of biological reality, right? And particularly if we're interested in capturing small molecules, we really do want to be able to do this, right? So AlphaFold 2, like I said, has no native support for non-amino acid entities. All that's beginning to change, and we might hear about that in the next in the immediate future. But you could use AlphaFold 2 structures for conventional docking, right? So you could simply take predicted structures, use them in a docking pipeline, and ask how well does that do, right? Alternatively, you could take aspects of AlphaFold 2, including its architecture, and essentially try to repurpose them for the task of docking. And I'll mention sort of stories on both of those ends. Next slide. So this was the task in which people were asking essentially how well do AlphaFold 2 structures work if we predict a model, if we predict a structure, and then task it with a docking sort of problem. On the left in blue is the variability in the ligand pocket site of a protein between different crystallographic structures of the same protein bound to different ligands. So these are different holoforms, and we're just asking how much variability is there in the binding pocket, and this is an RMSD on the y-axis, so fairly tight, not ton of variability. Then in orange is the same question, but just from AlphaFold 2 structures, right? And so what we see here is that AlphaFold 2 structures relative to the crystallographic structure seem to actually be quite good in terms of predictions, right? And then in green are the kind of standard homology models, which are sort of much higher up, so they look much worse. So on this level, right, this looks quite promising and suggests that in fact, AlphaFold 2 structures are sort of good starting points for docking. Next slide. However, once you actually do that process of docking into the AlphaFold 2 structures, what you see is, and so this is that was kind of flipped, where now is success rate, so higher is better. You see that crystallographic structures act as very good starting points for doing docking, but AlphaFold 2 structures, that's the ones in orange, they actually seem largely indistinguishable from green, which are the classical homology models. So that sort of improvement in accuracy doesn't really seem to translate into improvement in sort of docking scores. And it's likely that the reason this is the case is because the holoforms are not well captured by AlphaFold 2, and so we end up with a situation where it's sort of stuck on the apoform, and that's not really capturing the pocket where the binding occurs. Well, in the crystallographic instance, because we're using holo structures as the basis for docking, you do see that. Okay, next slide. I realize I'm sort of out of time, so I'll probably next slide. Next slide. Okay, I'll mention this slide now, basically, and there. So there has been now a lot of work on essentially repurposing AlphaFold 2 ideas and so on, or deep learning in general, to try to tackle the protein ligand problem. And there's been actually fairly exciting progress. But I just want to kind of provide this slide as a bit of a warning to people who use these methods. So what's shown here is essentially the performance of these various methods on the problem of docking, where here again the y-axis is success rate, so higher is better. And so we see that in the classical case, those are the bars on the very left. The models are actually not too bad if you provide them with the structure of the holoform protein, right? Now all the rest of the bars are ones from deep learning based methods. And what you see here, and this is what the kind of the warning, I guess, is that if these methods are applied sort of naively without any kind of energy minimization to sort of resolve physical pathologies, their performance is substantially inflated, right? So the kind of the lightweight, the light dashed versions of these bars correspond to kind of their naive performance, but then the solid versions of those bars are the actual performance if you account for the fact that they are introducing non-physical kind of pathologies into the structures. So in a way, what this is suggesting that a lot of these deep learning methods still have a long way to go in terms of being able to recover the proper small molecule geometry. And when not doing so, if one doesn't account for it, you actually get very inflated performance. Now this does, next slide, this does get sort of corrected if you do some energy minimization, and this is partly what this is showing, that kind of the purple is recovering that. So it's not sort of a completely hopeless, but still that delta between the physics space and the classical approaches actually does remain. Okay, if you please, you can proceed forward several slides. I'll just end on, you can move forward. Let me stop here. So I'll end with this slide. So as Christy sort of mentioned, we have this kind of OpenFold consortium, which is an academic industry collaboration with the intent of being able to essentially focus on problems in this field in a way that's steerable by industry interest, right? So we want to be able to kind of focus on questions that are of interest to folks here and elsewhere, and to do so by having a mechanism in which industry input can be taken into account when prioritizing what problems to go after. And these are some of the ones that I talked about today, and we're obviously open to input for more. So I guess just one more slide on the acknowledgements. So one more actually. So I'll stop here and just again kind of mention Christina, because she's really been very instrumental in kind of helping us get the OpenFold consortium up and going, as well as other folks in the OpenFold team who've actually done the work in developing these methods. Thanks for your time. Sorry for going over.